Good morning. Welcome from Kraków, Poland. My name is Marek Skowronek. I represent StatSoft Poland, and I would like to share my experience related to product quality monitoring, mainly in pharma industry. StatSoft Poland is part of StatSoft Europe and has over 30 years experience in advanced and predictive analytics and provides you with worldwide proven typical statistical software that can be used in highly regulated pharma industry and security relevant financial environments. In addition to typical statistical software, StatSoft provides services including consulting in all steps of your analytical projects, implementation of typical statistica into existing IT environment, adaptation of typical statistica to your needs, and setting up a business solution for you, and trainings on statistica itself, as well as product independent. Today's webinar presents the basic principles of implementing statistical process control and selected methods of data analysis, which can be used as a part of periodic quality review or ongoing process verification. In the second part of the webinar, a program dedicated to product quality monitoring will be demonstrated. Okay, let's start. The current changes and trends in the pharmaceutical industry have been defined by the International Conference of Harmonization. Recommendations outlined in guidelines Q8, Q9, and Q10 are now a point of reference to create new and update existing regulations. One of the important changes resulting from the ICH recommendation is to move away from the traditional perception of product quality as a compliance with the specification limits. Within this quality model, the waste is generated by products being outside specification. The traditional definition of quality assumes the reactive nature of pharmaceutical quality system, where actions are taken after out of specification event only. The definition of quality resulting from the ICH recommendation assumes a quality model that is a function of variability. In such a quality model, the waste is generated as a result of process departure from the target value. The greater the variability in the process, the poorer the quality of the product and the bigger the losses for both organization and patient. As a consequence, the modern definition of quality means proactive character of the pharmaceutical quality system, in which actions are taken before the occurrence of the out-of-specification result. An adverse event in this quality model is related to a special variability being called out-of-trend result. The principle of continuous process and product development is then fully applicable. What is the continuous improvement in modern quality model? It is about reducing organizational and patient losses by reducing variability, and it requires understanding the sources of variation and detecting the presence and degree of variation, understanding the impact of variation on the process and on product attributes, and controlling the variation in a manner commensurate with the risk it represents to the process and product. And control of variability requires the establishment of an effective process performance and product quality monitoring system covering every stage of the product life cycles and should include two steps: collection of process and product data, preferably in a real-time manner, effective transformation of data into information using, for instance, control charts, building knowledge about product quality and process by identifying the source of variability, and decision-making based on the knowledge gain. In 2015, the recommendation of ICH Q8, Q9, and Q10 have been taken into account in the new version of Annex 15 of Good Manufacturing Practice, introducing a validation life cycle that covers process design, process validation, and new requirement, continued or ongoing verification of process during routine production. ICH and G documents, while providing general principles of monitoring and knowledge management, do not describe how they should be organized and implemented in practice. Each manufacturer should therefore decide how to manage data and knowledge. So how to start building such a program? For instance, by answering the following questions. What kind of data should be collected and analyzed in order to add value for the organization and the patient? Two basic criteria should be taken into account: quality and effectiveness. Data should be collected for those parameters that are critical to safety, efficacy, and the quality of the product, and whose monitoring and trending is expected by pharmaceutical regulators as a condition for meeting the essential requirements of CGMP. Data should also be collected for parameters that are technologically important and whose analysis will increase the effectiveness of the technological process by reducing waste and shortening the production cycle. Next question. How should data be collected, stored to ensure the automation, integrity, and security? The basic principles of data integrity should be implemented, meaning the data should be attributable, legible, contemporaneous, original, and accurate. Next question. What statistical methods can be used to transform the collected data into knowledge in an efficient way? An effective monitoring program for process and product quality should include two interrelated programs. Experimental research conducted by R&D department, where mainly design of experiments methods are used to reduce the natural variability of the process, and observational studies conducted by the production or quality department, where statistical process control methods are used to identify special variability. Next question, how to communicate product and process knowledge so that it can be available anywhere at any time and for everyone? The sharing of data and knowledge can be realized in the following ways. By sending notification after identifying adverse events out of trend notification result, real-time process visualization by means of, for instance, control charts and quality metrics reflecting the performance of an organization, process, or system, and generating reports from the verification of the current process for the purposes of product quality review or ongoing process verification. The last but the most important question you need to answer is how implementation of an integrated knowledge management system will reduce the complexity and variability of the entire organization and increase its effectiveness. To address those questions, StatSoft provides solutions ranging from simple analytical add-ons, which can facilitate data analysis and reporting in various GMP areas. And advanced systems that enable collecting, analyzing, storing, and sharing information in all stages of the product life cycle for product quality monitoring, stability studies, and environmental monitoring. In 2018, with the support of the European Union Fund, StatSoft Polska started working on integrated system that will cover all stages of product life cycle, starting with research and development based on QBD approach, through validation of the process and continued ongoing verification of process during routine production. Now let's take a closer look at the basic of statistical process control. As per Annex 15, manufacturers should monitor product quality to ensure that the state of control is maintained throughout the product life cycle with the relevant process trends evaluated. Statistical tools should be used to support any conclusion with regard to the variability and capability of a given process. What is the main objective of process control? It's about process predictability. A process can be considered predictable if, on the basis of historical data collected, it's possible to predict how the process will behave in the future within the assumed variability limits. Predictable, stable process is characterized by random, natural variability, resulting from many cause and effect relationships, none of which is dominant in the process. In an unpredictable, unstable process, however, there are special causes and unidentified sources of variability that the process and generate the product being out of specification limits. But why should you care what type of variation is present? The answer is that the type of action you take to improve the process depends on the type of variation present. If the process has unpredictable adverse variation, the special cause responsible for the unpredictability should be identified and eliminated or compensated in some way. However, if the process has variability that is consistent and predictable, the only way to improve this kind of process is to fundamentally change the process, because there is no single dominant cause and effect relationship. A reliable way to separate common from special causes of variation are control charts. A typical control chart consists of observations collected in chronological order and three horizontal lines showing the central tendency and the upper and lower control limits, which include a band of natural process variation. As long as the points are randomly distributed within the control limits, the process shows a natural, predictable variability, and each subsequent result does not differ significantly from those observed in the past. Points outside control limits and or non-random points configurations may indicate a special cause, potential source of variability, and has significantly affected the critical attributes of the product. For statistical process control, which is based on control charts, to be effective and economically justified, four basic assumptions must be met. Foundation 1, 3 sigma limits. Foundation 2, estimation of sigma. Foundation 3, rational sampling and grouping. And Foundation 4, organizational culture. Control limits in the chart should be set at the distance of 3 sigma from the center line. Why? The 3 sigma principles, although having some theoretical background, was chosen by the mainly on the basis of empirical research. The control limits allow special causes to be identified and action to be taken in cases that are economically justifiable without generating too many false signals. These limits are also robust to violation of the assumption of normality of data distribution. even for individual observation, as shown in the table. Assuming data homogeneity, almost all points are contained at the distance of 3 sigma regardless of the distribution of data. Foundation 2, estimation of sigma. The process sigma should be determined as an intra-within sample average dispersion statistic. Why? The process sigma used to determine control limits should be a measure of natural process variability. Process estimation based on the average dispersion statistics calculated for each sample is an effective way to determine process noise. The choice of statistics used to determine the dispersion is not so important and may be the average of standard deviations or ranges or variances determined for individual samples. Foundation 3, rational sampling and grouping. A rational approach to sampling and grouping data should be applied when creating control charts. Why? The way in which data is collected and organized determines the questions to which control charts will answer. Sampling should be carried out in such a way as to minimize within sample variability, meaning representing the natural variability of the process, and maximize between sample variability, increasing the chance of detecting the occurrence of a special cause. A rational approach to data sampling and grouping should define what kind of sources of variability in the process contribute to within sample variability and which to between sample variability. For instance, within batch should have internal homogeneity, and the variation of interest might be batch-to-batch variation. Foundation 4, organizational culture. The introduction of statistical process control tools alone is not sufficient to improve the efficiency of the organization. Why? Control charts are as effective as an organization uses the knowledge resulting from the implementation of its statistical process control program. And this knowledge is only useful when it is the source of action. Hence, an effective statistical quality control requires the creation of quality culture, enabling systematic building of process and product knowledge throughout the product life cycle and continuous improvement of the process through identification, reduction, and control of sources of variability. To effectively use control charts, you must be able to interpret the control charts. How to do that? Start from visual examination of the chart. As a general rule, if a process is in statistical control, as in the diagram, most of the points will be close to the mean value and evenly distributed around the mean with no non-random patterns. Based on those assumptions, several quantitative rules have been defined and can be used to interpret what your charts is communicating to you. These rules help you identify when the variation on your control chart is no longer random and may result from the assignable cause. Let's start from zone rules defined by Nelson as listed in the table. The first test beyond the limits, one or more points beyond 3 sigma limits. It detects a large process shift away from the average. This is the main test in the statistical process control. Test Zone A, 2 out of 3 consecutive points in zone from 2 to 3 sigma or further. Detects a large process shift away from the average. It increases sensitivity without generating too many false signals. Zone B, 4 out of 5 consecutive points in zone from 1 to 2 sigma or further. It detects a small shift of the process from the average, increases sensitivity of the control chart without generating too many false signals. Test zone C, 9 points on the same side of the center line. It detects a small persistent shift in the process from the average, most commonly used for its simplicity. In addition to zone tests, there are non-random patterns tests listed in the table that may indicate special causes of variation as well. Test trend, 6 point increasing, decreasing, and it indicates a process that is trending in one direction. Stratification, 8 consecutive points outside zone C, above 1 sigma. May occur when you collect separate samples from multiple processes. Test over control, 14 consecutive points changing up and down is often due to over adjustment of the process. That is in statistical control, what may result in increasing the process variation. The next test, mixture, 15 consecutive points in zone C, below 1 sigma, may occur when you have multiple processes, but you are including all the processes in a sample, what may result in artificial widening control limits. Okay. Which run tests? Rule number 1, at the beginning, select points beyond limits. Identify and eliminate major causes in the process. Then consider additional configuration tests. Rule number 2, select up to 3 additional run tests, preferably zone tests that are process specific and whose detection is important for process improvement and economically justified. Rule number 3, for chosen configuration tests, define what they can mean for a given process and what action should be taken after detection of a given configuration. What type of control chart you can use in your statistical process control? The most commonly used chart is the individual and moving range control chart used for samples of size 1. It is a two-way chart that monitors the process location by means of individual chart and the process dispersion by means of moving range chart. The chart may be used to monitor critical attributes of the product such as assay, pH, impurity. The example illustrates the monitoring of assay in the finished product with one point beyond the control limit on both charts, signaling a potential special cause source of variability. Another method is X-bar and range or standard deviation control charts, which is used for sample size above 1. With variation estimated based on ranges or standard deviation in samples. It is two-way charts that monitors the process location by means of X-bar chart and the process range or standard deviation chart. The chart can be used to monitor the critical attributes such as dissolution or content uniformity. And the example illustrates the monitoring of the dissolution of a finished product with several points outside the location control limits while maintaining a high homogeneity of each batch on the R dispersion chart. This is a typical dissolution image. So how to handle this situation where most of the points are out of statistical control on X-bar chart? To solve the problem, use the three-way control charts where the standard X-bar control chart is replaced by two charts where the subgroup averages are treated as individual values with the limits calculated based on the moving range between subgroup averages. The chart can be used to monitor critical attributes, for instance, dissolution, in cases of high but natural variability in the location between batches, while at the same time high internal homogeneity of each batch on the spread chart. The application of this type of charts to the previous case allows the control limits to be extended, including all points. In case of parameters with different distribution than normal, which can result from their nature, for instance, parameters are physically limited, for instance, from the bottom, such as microbiology or acceptance value, a control chart according to the percentile can be used with a dispersion of the process estimated on the basis of the percentile of the theoretical distribution. It is a one-way chart. However, it should be noted that the Short control charts used in the process stability analysis are sufficiently robust to moderate violation of the assumption of normal distribution and do not generate an excessive number of false alarms. When to start calculating control limits? Rule number 1, calculate control limits after the process has been completed, including validation and subsequent commercial batches. Rule number 2, recalculate the control limits after each batch until data from 20-30 commercial batches baseline period is collected. And rule number 3, after 20-30 batches, freeze the baseline control limits provided the process has been found to be stable and capable. So when to change control limits? For instance, by answering yes to the following question. Do the data show significantly different behavior in relation to those collected in the past? And has the root cause for the change that affected the behavior of the process have been identified? And rule number 3, is the new behavior of the process desirable from the quality standpoint and expected to be sustained? What if the points are outside the control limits? Rule number 1, if the root cause has been identified, remove the point from the analysis and recalculate the control limits. Rule number 2, if the root cause has not been identified, do not remove the point from the analysis. And rule number 3, assess the risk of out-of-specification results at the release and the shelf life with impact and risk analysis if the out-of-specification risk is high for the points being out of control. Okay, so we have seen how control charts can tell you if your product's quality is consistent, but control charts do not tell you if the product is meeting specification limits. Once the process is in a statistical control, real effort at the process improvement can begin. Why? Because you can be sure that your process behaves in a predictive way. Process capability is one of the methods of measuring the effectiveness of a process in meeting specification, as well as measuring process improvement efforts. To determine that we can use capability charts that consist of histogram with control and specification limits shown. It allows to estimate to what extent the analyzed process defined by control limits is within the tolerance limits defined in the quality specification. You can also use numerical indicators of process capability. the most commonly used quantitative indicators in the analysis of process capability are CP, an indicator of the potential process capability, and the CPK indicator, which determines the current state of the process and takes into account the lack of process centering. The CP and CPK indicators are calculated on the basis of process sigma estimated from within sample variability, which defines the natural variability of the process. To evaluate the process, PP and PP process performance indicators are also used. However, they are determined on the basis of total process variability, including intra as well as inter-sample variability. So which one should you rely on? In case of processes being stable over time and centered to nominal value, all quality indicators have similar values. If your process is not in statistical control, CPK and PPK are meaningless because you cannot predict process sigma. So you have something to work on. It is recommended to report 4 indicators, CPK, PP, PPK, because they all contain information about the process. The difference between CP, PP, and CPK, PPK is a measure of the lack of centering of the process on the nominal value, and the difference between CP, CPK, and PP, PPK is a measure of process instability. The table shows recommended criteria for capability indicators. If indicator is above 1.33, attribute process meets specification limits in a consistent manner. No action is required. If the indicator is below 0.67, attribute process does not meet specification limits in a consistent manner. Take action. So when the capability analysis is reliable. Rule number 1, capability analysis is reliable for stable, predictable processes only. The data should follow normal distribution in case of standard capability indices, CP, CPK. Rule number 3, the sample size should be large enough, preferably over 100 to narrow confidence interval. Okay, so to sum up the step-by-step, the webinar. Step 1, select critical quality attributes, preferably 2 to 4 that are related to safety and efficacy of the product. Step number 2, collect data for selected critical quality attributes for extended period of time, preferably 20-30 batches to establish a baseline reference period. Step number 3, construct control charts for collected data and determine control limits for the future batches. Step number 4, develop control strategy, a methodology how to handle out of control event. Step number 5, implement control charts in routine daily work to evaluate each batch with respect to control limits. Step number 6, evaluate process stability using test 1, points beyond control limits, and additional 2 or 3 tests if justified. Step number 7, if the process has special causes of variation present, try to remove them using the control strategy defined. Repeat this step till process is under control. Step number 8, once the process is predictable, start to calculate the reliable process capability value, CPK, for instance. If the CPK value meets the goal, continue monitoring using the control charts. Otherwise, use experimental design techniques to reduce the natural variation and to improve the CPK value. Step 10, repeat the procedure till your process is stable and capable. But remember, when you reach this quality summit, treat it as a beginning of the journey, not the end. Okay, now let's take a look at the simple add-ons that can be used to analyze data collected during product quality review or ongoing process verification to generate, for instance, a statistical report. Okay, now we can switch to Statistica, and we have the data collected for 2 critical quality attributes, for instance, assay or impurity, from 26 batches. And we can analyze this data using product quality monitoring add-on, which is located in the pharmaceutical add-ons ribbon. Okay, we can open this program. And the first step is to enter the data into the program. So we can select this spreadsheet. And then we need to select variables to be analyzed. For instance, we can select assay. Then we enter the date of manufacture and the batch number. Okay, and this is all what is needed to do. And then we can generate the report with some default settings. Okay, the report has been generated, and let's take a look at this report. Okay, this is a statistical report and can be attached to the quality product review report or ongoing verification of the process. It contains descriptive statistics, raw data table with outliers table, and a run chart with raw data compared to the specification limits, and with the table that summarizes the out of specification results. The next section is histogram and basic statistic for the data collected. And the next one is a verification of the process model to check if the data follow normal distribution. This is a histogram and with a fitted normal distribution, and the verification of the normal distribution using quantile plot, and with numerical tests, Shapiro-Wilk normality test for them. So in our case, there is no sufficient evidence to reject the hypothesis HO. The data comes from normal distribution. The next section is dedicated to process stability analysis using typical control chart. For instance, in this example, it is individual and moving range chart with 1 point being outside control limits. And that may indicate a special cause present in the process. The next section is a section that you can check the run tests. You can take a look at non-random configuration of points. In this case, we have 2 out of 3 samples in zone A test. And the last section is about process capability analysis to check if the process is capable of meeting specification limits with capability indices, CP, CPK in PP, PPK, with its confidence intervals. This is a basic number of analysis that can be done for some critical quality attributes. And using this program, you can also select some additional tests, for instance, autocorrelation plot with parametric and non-parametric tests. You can also select the verification of long-term trend, linear trend with parametric and non-parametric tests. And you can use also control charts based on percentiles or cumulative sum control charts. So the content of the report depends on you, as well as some settings also can be changed according to your internal procedure. For instance, for CPK, PPK limits. And when you are satisfied with your control limits, you can calculate those control limits and save into the spreadsheets. So next time you can enter the data and compare the next subsequent data to the baseline control limits established based on the reference batches. Okay, so when it comes to the add-ons, that's all. And if you have any question related to the statistical process control analysis or to the software, please send me an email. And I would like to thank you for participation in this webinar, and thank you.